Hello, everyone. We are going to jump right into our biomolecules lecture for today. As always, you should be filling in your biomolecules note sheet with the terms that are present on these slides. So before we can even talk about biomolecules, let's review the carbon atom. Most biomolecules are made up of carbon atoms bonded to other atoms, but carbon makes up the majority of these biomolecules. So carbon has an atomic number of 6, which means carbon has 6 protons in the nucleus, as well as 6 electrons surrounding the nucleus in what's called the electron cloud. Remember, there are 2 electrons in the first level, or the first electron shell, and then there are 4 valence electrons, which means there are 4 electrons in the outermost shell of the electron cloud. Carbon, which this means, so 4 valence electrons means carbon can bond or form up to 4 covalent bonds with either other carbon atoms or other elements in order to form molecules. So, because carbon can form up to 4 bonds, these large biomolecules often have these long chains of carbon. So like I just said, carbon atoms can form single, double, or triple covalent bonds with other carbon atoms in order to form these really long chains. So I have given you a couple pictures of these carbon chains right here. These are referred to as carbon skeletons. So carbon skeletons are any variation of carbon atoms bonded together. So you can have a straight carbon chain, a straight skeleton, a branched carbon skeleton, or a carbon ring. So we will look at carbon rings when we look at sugar molecules or carbohydrates. Each line between the atoms, so for example, each line between the carbon atoms or the carbon and the hydrogen represents a covalent bond. And what's really important to note here is that energy is stored in these bonds. So when we break these bonds, energy is released. Chains of carbon can take on millions of large and complex structures. So these are relatively small carbon skeletons, but they can be very, very large. So now that we've talked about carbon, we reviewed carbon and carbon skeletons, we are now going to talk about biomolecules. So biomolecule is also referred to as a biological molecule, that's the longer version of the name, or a macromolecule. Those two terms are synonymous. So macromolecules or biomolecules are large and complex molecules that are present in living organisms, and these molecules are essential to life. We cannot function on this earth without biomolecules. To form biomolecules, they undergo a process of polymerization. So polymerization is the building of large molecules from smaller ones. We've seen these images before. So to build a biomolecule, you have to form bonds between what are called monomers to form a polymer. So this is the process of polymerization, forming a polymer or a larger molecule from smaller molecules like this. Each of these lines here represents a covalent bond. So monomers, think of mono. When you hear mono is single. So monomers are simple molecules or a small unit. Chemical bonds form between monomers to form polymers. So polymer, poly means many. A polymer is a macromolecule that consists of repeated linked monomers. So monomers bonded together is a polymer. Monomers are usually linked together by covalent bonds. And polymers are chemical bonds that form between polymers form macromolecules or large biomolecules. So a macromolecule or a biomolecule is a large polymer. So basically, if you're looking at the scale, monomer is the smallest. You bond monomers together, you get a polymer. You bond polymers together or you fold them together in a certain way, you get a macromolecule or a biomolecule. So to form a polymer from a monomer or to form a macromolecule from polymers, you have to undergo the dehydration reaction. So the dehydration reaction is a chemical reaction that removes water molecules in order to form a polymer. So for example, you actually completed this, you modeled this yourselves, but here is a formal model. We have a 3-carbon polymer right here, and then we have a monomer, right? So we want to link these together to form a larger polymer. What we're going to do is link the OH from one monomer and the H from the other. Remember, OH plus H is H2O. So once we remove H2O, we are able to bond this monomer to the short polymer and get a 4-carbon polymer. This is an example of a dehydration reaction. You're removing water and linking monomers or polymers together to form a larger molecule. So Now that we understand how monomers form polymers, polymers form macromolecules, and we understand a little bit more about how biomolecules are formed, let's talk about the biomolecules themselves. So there are 4 biomolecules in total. The first is carbohydrates. Carbohydrates are organic molecules, which means they're made up of carbon. And carbohydrates themselves are made up of carbon, hydrogen, and oxygen. All right. So the function of carbohydrates are, number 1, is to store energy. So carbohydrates are a source of energy. When carbohydrates are broken down by organisms, they can obtain energy. For example, we know that potatoes are an example of a carbohydrate. When we eat potatoes, we are able to digest the potato and break it down. As that potato is being broken down by our cells, we are able to release, or we are able to obtain energy from that potato. So carbohydrates are a source of energy. They're also important structural materials. So when you see the word structural, think of structure. Carbohydrates form these structures of various cell parts and also in plants. So carbohydrates form the structures of most plants. So here I've given you a little cheat. The structure of carbohydrate is monomer, polymer, macromolecule. The monomer of a carbohydrate is called a monosaccharide. This is a sugar, a simple sugar, a one sugar molecule. There are 3 monosaccharides that I want you to focus on: glucose, fructose, and galactose. Just remember, you do not have to memorize these structures. I just want you to see how they are all simple sugars. They are all one sugar molecule. Glucose is the middle one right here. We will be looking at glucose a lot. Glucose is the foundation for a lot of biological processes going forward. Glucose is an example of a monosaccharide. Next, we have disaccharides. So disaccharides, di means 2. Disaccharides are made up of monosaccharides that are covalently bonded together. So when you put 2 monosaccharides into a dehydration reaction, you remove water. So for example, see here you have an OH from one fructose and the H from the other. You remove water, you get a disaccharide. So these 2 monomers are undergoing the dehydration reaction, removing water, we get a disaccharide. This is a covalent bond between the 2 monomers. An example of a disaccharide is sucrose or table sugar. So the sucrose, this molecule right here, is an example of glucose and fructose bonded together to form a disaccharide. Finally, when you form multiple bonds between many monomers or many polymers, you get what's called a polysaccharide. And polysaccharide is a macromolecule of carbohydrates, which is made up of 3 or more monosaccharides bonded together. So examples are glycogen, starch, and cellulose. Glycogen is a storage polysaccharide. So glycogen is a storage polysaccharide in animals, which means that the glycogen is responsible for storing glucose in our cells. So when, for example, when our blood sugar levels get low, or when glucose in our blood runs low, glycogen is responsible for breaking down that stored glucose. So glycogen holds glucose. Our body can break down the glucose in glycogen and release it into the bloodstream. You can typically find glycogen in liver cells, but also in muscle cells as well. So for example, we use our muscles to run, to lift, to really carry out all of our functions. We need extra glucose. We need extra energy in those cells so that we continue to move. Starch is a carbohydrate or a polysaccharide. Starch is responsible for storing glucose in plants. So like humans and animals have glycogen, plants have starch. So potatoes, corn, rice, they are all starches. They store glucose. Humans often rely on starches for a source of energy to replenish their glycogen stores. And finally, you have cellulose. So cellulose is a structural polysaccharide. And it's what gives plants their strength and rigidity. So it's a polysaccharide that is responsible for giving plants their strength. So for example, wood and paper is made up of cellulose. So starch and glycogen are storage polysaccharides. They store glucose. Cellulose is structural, which means it gives plants its structure. The next biomolecule we're going to talk about is nucleic acids. Nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And the function, carbohydrates have one main function, and that is to store and pass on hereditary information from parent to offspring. All right. And unlike carbohydrates, nucleotides are made up of monomers. And then when you bond monomers together, you get a polymer or a macromolecule. So nucleotides are the monomers of nucleic acids. All right, so when you bond nucleotides together, you get nucleic acid. Nucleotides contain one sugar, one phosphate group, and one nitrogenous base. And they are joined together by the covalent bond by the dehydration reaction. So dehydration reaction joins nucleotides together, and that forms our nucleic acids. There are two major nucleic acids. So there are two nucleic acid polymers, DNA and RNA. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid, and the two differ by the difference of sugar. So DNA has deoxyribose as its sugar, and RNA has ribose as its sugar. Really quick, this is an example of a nucleotide. This is a simplified model of a nucleotide, but it is a good representation to show that every nucleotide has a sugar, a phosphate group, and a nitrogen base. Here is a little more bit of a complicated structure of a DNA molecule, and you can see that these are the sugars, these orange hexagons, these yellow P's that you see here, these are your phosphate groups, and then your pink, purple, red, and green hexagons are your nitrogenous bases, which together they all form DNA. Your third biomolecule is protein. So proteins are made up of carbon, hydrogen, oxygen, and nitrogen. And proteins have several different functions. They are really important for the functioning of the cell. Most cells are made up of mostly proteins. So for example, some of the functions include controlling the rate of chemical reactions, regulating certain processes in the cell, forming important structures, so they're structural, and then they're also really important for transport of substances into and out of cells. We are going to focus on proteins a lot in the upcoming units for cells and things like that. Proteins, their structure is monomer, then polymer, then macromolecule. So the monomer of a protein is called an amino acid. All right, so when we have, this is an example of amino acid. Amino acids have an amino group, a carboxyl group, and a side chain. Remember, every side chain is different for every single amino acid. So there are 20 total amino acids. They all have amino groups, carboxyl groups, and a hydrogen. They all have different side chains. All side chains have a different structure. And these side chains are important because they help amino acids carry out specific functions. So these side chains really change the entire structure of not only the amino acid, but the protein itself. The polymer of a protein is called a polypeptide, and that is made up of 3 or more amino acids bonded together. And when you bond these amino acids together, so all of these little beads on this chain right here, each bead, each color represents a different amino acid. When you bond these amino acids together, we get what's called a polypeptide. All right, and these polypeptides can then form together. They can bond together and fold together into these 3-dimensional shapes and form a protein. So each bead, let me reiterate, each bead on this chain is an amino acid that are covalently bonded together. When you bond them together, you get a polypeptide chain. This is the polypeptide chain. If you fold multiple polypeptide chains together, you get a protein. Again, protein structures are really, really complex, and these structures determine their function. If you change the structure or if you change the surrounding of the protein, you may negatively impact the structure of the protein and make it impossible for the protein to carry out its function. So really, if you change, for example, the amount of salt in the environment of the protein, that could make the protein non-functional and it will no longer carry out its job. Here is a photo that just shows the monomer, polymer, and macromolecule in a simplified form for proteins. And our last biomolecule is lipids. So lipids are made up of carbon, hydrogen, and oxygen. And the function of lipids, there's 2. Well, there's actually more than 2, but we in this class are going to focus on 2. Number 1 is to store energy. So lipids are a really, really good source of energy. And number 2, they make up what's called the cell membrane. So that is a part of the cell. Here is an example of what a lipid looks like. Take a look. You only see carbon, hydrogen, and oxygen atoms. So lipids have 2 parts. So their monomers make up 2 parts. One is called a glycerol and one is called fatty acids. So a glycerol is a 3-carbon molecule. There's 3 carbon atoms and it's bonded to oxygen. So this is a simplified form. Glycerol is up here in this molecule. Glycerol, is represented in the blue box. So it's 3 carbon atoms. All right. Covalently bonded to the glycerol are called fatty acids. So we also call them fatty acid tails. These are non-polar hydrocarbon tails. So that means that, fatty acids, these three tails are called fatty acid tails. So these three long chains of